Welcome to this explainer. We're going to just jump right in today because we're looking at something that is, quite frankly, one of the most mind-bending realities of cellular biology. We all heard the classic high school line, right? The mitochondria is the powerhouse of the cell. But that incredibly simple little phrase is hiding a literal biological thriller playing out inside your body right at this very second.
So what if I told you that alien DNA is operating inside your cells right now? I know, it sounds totally like science fiction, but it's actual biological fact. See, mitochondria are completely unique compared to all the other organelles floating around in your cell's cytoplasm. Why? Because they pack their own distinct DNA. It's an entirely separate genetic code from the human nuclear genome that makes you, you. So how on earth did this alien code get inside us?
Well, the answer to this mystery is a process called endosymbiosis. The scientific consensus, which is heavily backed up by some really rigorous DNA sequence analysis, is that mitochondria actually evolved from ancient bacteria. Eons ago, these bacteria set up a symbiotic relationship where they literally moved in and lived inside larger host cells. And here's the wild part. When scientists sequence modern mitochondrial DNA, they find these striking similarities to a present-day bacterium called Rickettsia prowazekii. Rickettsia is an intracellular parasite that, exactly like our mitochondria, can only reproduce when it's inside a eukaryotic cell. It's basically the ultimate evolutionary roommate situation.
Here's our roadmap for this explainer. First, the alien inside our cells. Second, powering the cellular machine. Third, a rebellious genetic code. Fourth, importing parts and assembly. And finally, number 5, mutations and disease.
All right, part 1. The alien inside our cells. Meet your resident bacterium.
What's super fascinating here is just the sheer variety of mitochondrial DNA across different species. It exists in multiple circular copies, exactly like bacterial DNA. Now, the human mitochondrial genome, it's tiny, really tiny, only about 16 kilobases. Plants, though, can have genomes well over 200 kilobases. Take Arabidopsis, for example. It sits at a whopping 367 kilobases. But if you want to see the real bacterial ancestry, you've got to look at the protozoan Ricklamonas americana. With 97 genes, it looks incredibly similar to that ancient bacterial ancestor, before it started tossing out its genetic baggage.
Moving on to Section 2, Powering the Cellular Machine, Anatomy of the Power Plant.
So, how does this domesticated bacterium pay its rent? By generating the vast majority of our usable cellular energy. But to understand how it pulls this off, we have to look at its architecture. Mitochondria are wrapped in a double membrane system, and the contrast between the two layers is night and day. Imagine the outer membrane as a freely permeable chain-link fence. It's got these proteins called porins that act as open channels, letting small molecules just drift right on through. That means the space just inside this outer fence is pretty much identical to the regular cellular cytosol. But the inner membrane? That is a highly restricted, impermeable, high-security vault. It's absolutely packed with proteins, over 70% of its composition actually, and it's folded up into these complex structures called cristae to massively maximize its surface area. The fact that this inner vault is totally impermeable is absolutely critical.
And why is it so critical? It all comes down to oxidative phosphorylation. First, you've got your raw fuel, pyruvate from glucose and fatty acids, and these get imported right into the deepest part of the mitochondria, the matrix. There, they're converted into acetyl-CoA. Next up, the citric acid cycle breaks that down into carbon dioxide, capturing the energy as high-energy electrons on molecules called NAD+ and FAD. Finally, these electrons are handed off through a chain of carriers embedded in that high-security inner membrane, passing them all the way to oxygen. This transfer literally pumps protons across the membrane, building up an electrical charge. Because that inner membrane is completely sealed, those protons are trapped, and the mitochondrion harnesses that massive potential energy to crank out ATP, the ultimate energy currency of our bodies. Section 3. A rebellious genetic code. The organelles' secret language.
13. That is the exact number of proteins actually encoded by the human mitochondrial genome. Think about that for a second. Over billions of years of evolution, this organelle has offloaded almost all of its original genetic information to the human host's nucleus. It has stripped its own personal code down to the absolute bare essentials, just 13 proteins that are strictly necessary for the electron transport chain and oxidative phosphorylation, plus a handful of RNAs to translate them.
Now, almost all of biology relies on a standard universal genetic code. Normally, a cell needs at least 30 different transfer RNAs, or tRNAs, to read the mRNA codons and build a protein. using a standard wobble rule for matching bases. But our human mitochondria? They only encode 22 tRNAs, so how on earth do they manage? Well, they use an extreme wobble. Basically, a single U base on the mitochondrial tRNA can pair with literally any of the 4 bases on the mRNA. It's a biological hack that lets a single tRNA recognize 4 different codons. And if that wasn't rebellious enough, some specific codons actually translate to completely different amino acids in the mitochondria compared to the universal code. It is quite literally speaking a secret language right inside your cells.
Okay, Section 4, Importing Parts and Assembly, the logistical nightmare.
Since it only makes 13 proteins itself, over 95% of the proteins a mitochondria needs are actually coded by your nuclear DNA, manufactured out in the cell cytosol, and then somehow they have to be shipped into the organelle. So how do they get past security? They use pre-sequences. You can think of these like VIP passes tacked onto the front of a brand new protein. They're these specific little tags of 20 to 35 positively charged amino acids. These pre-sequences dock with receptors on the surface of the mitochondria to kick off an incredibly complex, highly coordinated import process.
Here's how this logistical nightmare plays out, step by step. First off, a protein can't be shoved through a membrane while it's fully folded up, so a molecular chaperone called cytosolic HSP70 unfolds it. Then, the VIP pass gets scanned by the molecular customs agents. First is the TOM complex, which stands for translocase of the outer membrane. Once it clears the outer membrane, it hits the TIM complex at the inner membrane. Now, remember that electrical potential we talked about earlier, the one generated during energy production? Because the matrix inside is electrically negative and the protein's VIP pass is positively charged, the mitochondrion literally uses its own electrical power grid to physically pull the protein inside. Finally, another chaperone, matrix HSP60, refolds the protein back into its functional shape. And, as you might guess, it burns ATP at every single step.
Of course, not every protein goes all the way into the deepest part of the matrix. Some of them have stop transfer sequences that basically hit the brakes halfway through, embedding the protein perfectly into either the inner or outer membrane. Oh, and it's not just proteins, by the way. The lipids making up the membranes themselves have to be imported from the cell's endoplasmic reticulum. But the mitochondrion does keep a few tricks up its sleeve. It synthesizes a completely unique lipid called cardiolipin right inside its own walls. It's this bizarre double phospholipid with 4 fatty acid chains, and you will barely find it anywhere else in the entire cell.
Which brings us to Section 5, Mitochondrial Mutations and Disease, when the alliance falters.
To really grasp the impact of mitochondrial disease, we have to look at how we inherit this organelle in the first place. You inherit your mitochondrial DNA entirely from your mother, no exceptions. When an egg is fertilized, the sperm does not contribute its mitochondria to the embryo. What this means is, there's a direct, maternal germline inheritance of this alien DNA, passing straight from mother to child, generation after generation.
Because of this highly unique inheritance pattern, if a mutation pops up in the mitochondrial DNA, it is transmitted maternally. And unfortunately, these mutations can be absolutely devastating. Take Leber's hereditary optic neuropathy, for example. It's a condition that leads to blindness, and it is caused directly by mutations in the mitochondrial genes that encode the parts of that electron transport chain we broke down earlier. When that energy grid isn't firing at full capacity, highly demanding cells, like the ones making up the optic nerve, just simply die off.
I want to leave you with a pretty profound thought here. As we live, as we breathe, and produce energy every single day, our mitochondrial DNA is progressively accumulating mutations over our lifetime. A lot of scientists are now suggesting that this progressive decay might actually be a primary driver of the aging process itself. So, think about it. Does the secret to human aging lie not in our own nuclear DNA, but in the slowly decaying genome of an ancient bacterial invader that just decided to take up residence inside us billions of years ago? It's a wild, fascinating perspective on what it actually means to be human. Thanks so much for joining me for this explainer, and keep exploring the incredible science operating inside you right now.